feature of this microscope when compared to light microscope. Also, here the light source is electrons. Electron is the light source for scanning electron microscope, whereas in light microscope, that's a bright field light source. So once when the electron beams strikes the samples, both the electron and photons are emitted. That is the signal emitted. If you have an appropriate detector, then you can get the information you want. So last class we discussed that is the electrons, the tungsten filament emitted the electrons. It's a pass through the column and hit the samples. So once the electron beams strikes the samples, it's emitted different signals. One is a primary backscattered electrons, and cathodoluminescence, secondary electrons, and agar electrons, X-rays, specimen current. So if you get in the primary backscattered electrons, you can get atomic numbers as well as topographic informations. If you detect the secondary electrons, you can get the topographical informations. Cathodoluminescence is, you will get detailed, electrical information of the samples. Also, the agar electrons, the surface is sensitive, and also the compositional informations you will get. The X-ray signal through thickness, you will get the composition information of the samples. So this is, the electron microscope is not only just you see the visualize the topography or the surface of any samples, but also you can get many informations like electrical information, compositional informations, how thick that is, and the atomic number you will get. Also the electrical informations of the detailed structure. All this data you will get it from this electron microscope. So how does this electron microscope works? So the electrons are generated in the electron gun. Tungsten filament serves as the source of our electrons. Once the electrons, the tungsten filaments, generated electrons will be focused in front of the anode. So to move the electrons, down the column, the voltage difference between the tungsten filament and the anode, will calculate it. Based on that, you will determine how much, electrons is striking the samples. This voltage differences between the tungsten, filaments emitted as well as in the anode, these differences we called the accelerating voltage and can be varied between 0.2 to 40 kV, which determining the energy and wavelength of the electrons within the beam. So the beam of electrons will be, condensed and focused as a fine spot on the specimen by 2 to 3 electromagnetic lenses located in the microscope column. So that is another important, like when the electrons emitted by the filaments, then it is, go through the column. It's a protected shield and striking the samples. So in between, when the travel of the electrons is condensed and focused, that is most important thing. Otherwise it will be dispersed. so that you will not get enough electrons striking the samples, that will end up with, you cannot get it the detailed structure. So the electromagnetic lenses that will condense and focused as a fine spot in the column. So the two main functions of the first two lenses, that is a condenser lens and are to control the beam current, that is number of electrons striking the specimen, and also the final size of the area illuminated on the specimen, the spot size. So once the electron striking the samples, the area where the electrons is striking is important. How far it will striking, so that you will see wherever the electron striking, that area of samples will be you can see the detailed structure. So we call that is the spot size. When you increase the spot size, the electron will be dispersed, so that you will get more area to visualize. But when you reduce the spot size, it's narrowed down the electrons, so that more electrons is striking a small area, so that you will see which increases the brightness and contrast, which gives you more detailed structure when compared to the spot size is increased. The third condenser lens will be used primarily to focus the beam of electrons on the surface of the specimens. So the interaction of the electron beam with the sample or specimen various signals, that is the secondary electrons, backscattered electrons, X-rays, are produced and can be detected. The signals from the interaction between electrons and the samples are used to obtain the information of the sample, either topographical, compositional, or elemental, or structural images, which all depends on which signal is used. So that's the most important thing, then the interaction of the electrons beams with the samples. So what kind of signals you wanted to get? Based on that, you will detect them, or using either, if you need just topographical or compositional or elemental or structural images, which all depending on which signal is used. So the secondary electrons is generated from the collision between the incoming electrons and the loosely bonded outer electrons. So that is, the low energy electrons, we called it is. So the secondary electrons which generated close to the surface is escape. That is, like once it go and strike the samples, and then it will be bounced back, so that you will get detailed, the topographic informations. So the number of secondary electrons, which are always greater than the number of incoming electrons. So the backscattered electrons is a fraction of incident electrons is retarded by the electromagnetic field of the nucleus, and if the scattering angle is greater than 180 degree, the electron can escape from the surface, so that you will get a detailed, topographical informations. The X-rays are photons, it's not an electrons. Each element has a fingerprint X-ray signal. The poorer special resolution than backscattered electrons and secondary electrons, which are relatively few X-ray signals are emitted, and the detector is inefficient. It's a relatively long signal collecting times are needed when you use these X-ray signals. As we see, it's a low energy electron, the secondary electrons, high resolution. You can get the surface signal which dependent on curvature. So the backscattered electrons is mainly high energy electrons, bulk signal which depend on atomic numbers. The X-ray is a longer recording time needed. When a scanning electron microscope is used, the electron optical column and sample chamber must always be at a vacuum. So as we see, the vacuum is very, very important in the electron microscope because the column, electron optical column and the sample chamber must be important, because if the column is in a gas-filled environment, electrons will be scattered by gas molecules, which would lead to reduction of the beam intensity and stability. That's one reason. Other gas molecules which could come from the sample or the microscope itself could form compounds and contents on the sample. This would lower the contrast and obscure detail in the image. So, why do we need then the vacuum? It's a chemical, and the thermal stability is necessary for a well-functioning filament. A pressure a field emission gun requires the 10 to the power of 10 torr, and LAB6 is a 10 to the 4th. There's no need to worry about all these details, but our main goal is, why do you want to have a vacuum? It's one reason is a corrosion if it's not vacuum. Also, it requires gives you the thermal stability for well-functioning filament. Also, the signal electrons must travel from the sample to detector. Vacuum requirements is dependent on the type of detector. So the resolution is very, very important for electron microscope, because here we are going to increase the higher magnification, like more than 500,000 times magnification, so that the resolution is also important when you increase the magnification. So the best resolution that can be obtained, the size of the electron spot on the sample surface. As I mentioned earlier, that the area of electrons striking is very, very important. If the more area, if you increase the spot size, that means it's more area cover, but it disperses the electrons, so that the resolution will be reduced. So the spot size is reduced to small area, then you will get more resolution. The volume from which the signal electrons are formed defines the resolution. So the scanning speed, another important is the scanning speed. So vacuum is important, the resolution is important, and the scanning speed. So the scanning speed means the electrons striking the samples and bounce back. So these two is important. How fast the electrons striking and how fast the electrons bounce back. So a weak signal that requires a slow speed to improve signal to noise ratio. When doing a slow scan, that is the striking and bounce back, the electrons that drift in the electron beam can affect the accuracy of the analysis. So that is most important. If the scanning, back and forth is optimal, is very, very important. So far we discussed about the scanning electron microscope's basic principles. So why do we want to use a scanning electron microscope, and how important it is, and the basic structure of the the components of electron microscope, and what are all the factors that influence the quality of your images, the resolution, contrast, and everything. So now we understand that the scanning electron microscope is important to detailed structure of external surface, that is a topography. And top of that, you will get a lot of other signals that will help you to get more detailed, not only the structure, but also electrical current, compositional elements, and atomic numbers, X-ray details. So these kind of informations will help to get more data on your samples. So to achieve this, we need to have a samples process. That's most important thing. One is, as we discussed previously, like fixation is first fundamental, informations you need to understand. So fixation, dehydration, embedding, then here we don't have any sectioning, we don't have staining. Instead of the staining, coating your sample, then observation. So here the basics is 4 steps. So one is fixation, dehydration, the critical point drying, that is to dry the sample before you coat the sample, and then mounting and coating. So this is a 4 important steps for scanning electron microscope. The specimen at a high vacuum requires a sample fixation and dehydration or freezing. So that is important. You have to specimen at a high vacuum that requires sample fixation and dehydration or freezing. Also, the charging is minimized by coating sample with metal or carbon or lowering the operating kV. So when you work on this sample, as we discussed, the vacuum is important to prevent the corrosion, and which helps the electrons travel to the striking the samples. Is also here the charging is minimized by coating sample with metal or carbon. So that is another important thing, we have to coat the samples. So specimen at a high vacuum requires a sample fixation and dehydration. So that is another interesting feature of this process. So here, when we go with this diagram, when you look at this left side, so specimen, you have a specimen. So for scanning electron microscope, it does not, whether it is, live cells or tissue samples or, biological samples. Any sample you can process for scanning electron microscope. So here that, 2 different or 3 different ways you can process this sample. So first sample is, if you have a sample that you wanted to visualize to detail the structure of the sample with the scanning electron microscope, if the material that has, any tissue sample or any other bacteria or any biofilm, so you wanted to see this kind of, for example, any metals or any implants or anything, you wanted to see what is going on with the coating the samples on the surface of the, material. If it is just metal or it's not living material, like wood or metal or any object, you no need to fix the sample. You can just directly coat the sample. So when you look at here, you directly, if you have a sample that does not require fixation, that means there is no, biological materials. That's any, things that you can directly mounting on the stub. That is to hold your sample. You no need to fix or no need to dehydrate or drying, because already the wood, if you really, for example, wood, you no need to do anything. For metal, you no need to do anything. But your goal is not only just the metal or not only just the wood or any material, but you wanted to study on the surface. If you wanted to study any biofilm, any bacteria forms biofilm on the surface of the wood or the metal or any implants or any material, then it requires a fixation dehydration, because there is a bacteria or other tissues, cells are grown. So that require fixation, because the bacteria has to fix. You then remove the water content, and then you can dry the samples, and then you can coat the samples. So now you understand that the same material, but how you are choosing different things. That's the most important thing. So if there is no any samples with the living things, then no need for fixation, dehydration, drying. Directly you can mounting the samples. If the sample contains any other living things, then you have to fix the samples with the glutaraldehyde, or you can just fix with the osmium tetroxide, and then you can dehydrate the sample. Once you dehydrate the sample, then you can dry the sample, and then you can mounting on the stub. The stub is to hold your sample, and then coating. Also, you have a sample with the freezing. If you fix your sample with the freezing and also liquid nitrogen, then it require dehydration, then mounting, and then coating and observation. So this is a 3 different approach, right? The scanning electron microscope, you can able to process your samples. So as we know that the fixation is always, we described several times with the glutaraldehyde or osmium tetroxide, you can fix the sample, and then you can dehydrate the sample is either with ethanol or methanol, the concentration from lower concentration to high concentration, like a 25%, 50%, 75%, and 100%. So the purpose of this critical point drying is to dry the sample. So that means already when you dehydrate, the final step is 100% ethanol. So now you cannot take the sample directly to coat the sample. If there is a ethanol, that will not coat, right? So the coat means it's a gold coat or carbon coating. So that's why the critical point drying is important. The purpose is to completely dry specimens for mounting while maintaining the morphological details. So now the ethanol is replaced, by water. So first is fixation and then dehydration. So during the period, now we remove the water and replaced by ethanol. So the next step is the ethanol, exchanged for liquid CO2. So now we are replacing the ethanol by liquid CO2. So this critical point drying instrument, that is carbon dioxide brought to critical point, that is 31.1 C, and the pressure is 1000, which becomes a dense vapor phase. So the gaseous carbon dioxide vented slowly to avoid condensation. Then the dry sample is ready for mounting. So that is interesting is previously for light microscopy or fluorescence microscopy or laser capture microdissection, during this period, we did not do dry the sample. The dry the sample is only the last step. After you transfer onto the slide, you stain it, and later only we dry the samples and observe. So here also, the sample is, we are not taking any sections. We just processed whatever samples that, and we can use to visualize the surface of the samples. Then the next step is coating. So next step is the alternate approach, that is the freeze drying. That's another important one. So here also the sample, if you fix the sample with the liquid nitrogen cryofixation, then the sample should be, after fixed with liquid nitrogen, placed in a vacuum evaporator on cold block. That is approximately minus 190 degrees C. Then left under vacuum for several days to sublimate water. Then it is ready to go for mounting, and then coated the samples. So the last stage of this, one step ahead, like the mounting the specimen onto the stub. You remember that light microscopy, after you take a sections, you transfer the samples onto the slide, glass slide. So the glass slide is, the slide is holding your sample. Whereas in electron microscope, scanning electron microscope, you processed your samples and then you wanted to hold your samples, right? So that is the, part is called the stub. The stubs are specimen holders, which is specific for the instrument being used. So each instrument have their own, stub. The specimen is held to stub by conductive tape, paste, or glue. So you wanted to, hold your samples, something to hold, right? So you can use glue or a glue tape that will have a both, adhesive on the both sides, so that one is stick onto the stub, the other, the top portion of the tape, that is also glue. You can hold your samples. You put the samples under, or we have a glue also. You can paste it, and then you can place the samples on the stub, so that will hold your samples. So now ready to go for coating your samples. So why do you have to coat your samples with either carbon coat or gold particles? This is to cover the surface of your samples. So your main goal is you are going to measure the surface of your samples, what's the compositional, how the electrical, and then on top of that is the feature of samples, the morphological features. So for that, it requires the coating your samples. One is, when you coat the samples, that is, it's a spray, the gold, the gold is particles spray on the surface of your samples, which increase the conductivity of electrons. if the gold or carbon, when electrons is striking the samples, if you coat the samples, that will conduct the electrons uniformly and then bounce back, so that you will get good structural views. So that is, it gives the increased conductivity and also reduce the thermal damage. So if you coat the samples with the carbon or gold particles, that will give you some deposit, a thickness that gives you thermal damage when the electrons are striking the samples. Also, the signals, when you're getting from the electrons striking your samples, the interaction between the electrons and the samples, if you coat your samples, that increase the secondary and backscattered electron signals. Also, electron emissions. So the electron emissions, that is, the electrons are, ejected from the surface of any given samples. That will be another important thing, that is stimulated by temperature or electric field. So that is also very, very important, that when you coat your samples. Also, the mechanical stability, like because it deposited, like you coated the surface with the gold particles or carbon, that also mechanical stability for your samples. So these 5 important points, that is to consider why you need to coat your samples. Increased conductivity of electrons, reduction of thermal damage due to the electrons striking the samples frequently, and also increased the secondary and backscattered signals due to when you coat your samples. Also the electron emissions, that is when you striking this and emitted, that is also increased, and mechanical stability. So these are all very, very important when you coat your samples. This is like similarly when you stain your samples in light microscopy. So accomplishment, accomplished by, how do we success with this, is fixation. If it is a biological samples, you have to fix your samples, preferably osmium tetroxide or glutaraldehyde. Also, as discussed, when you cold your samples with the stub, you have to use glue or painting. So the sputter coating, how we are coating the samples. So there are several ways to coat several gold or gold palladium target. So that is, it is also a small instrument, and vacuum also built approximately 2 millibar. And also the coating, the thickness of the gold particles emitted on the surface of your sample, the thickness should be 7.5 mm to 7.5 nanometer to 30 nanometer thickness, so that will be coating your samples. If you increase the, sputter coating, that will give you more stability. But, if you go for too thick, the features, the structural topography will be reduced. So optimal thickness of coating your sample is good when you see topographical images. We also use the carbon coating for samples. Not all the material you can coat the samples. Though, for samples in SCEM where the X-ray informations, if you're working on that, then it requires the carbon coating instead of gold particles. It also helps the replicas. It works under the vacuum. Also, carbon rod may need outgassing during this process. So, it's just warning that do not look directly at heated electrons in this coating your samples. Whereas in the gold particles, it's a small chamber with vacuum, so it doesn't give you any harms to things. So far we discussed about the scanning electron microscope and the process and things. So what are all the, why do you need to use this scanning electron microscope, and what are all the features you can able to see, and does it require all the samples should go for scanning electron microscopic observation? So some of the experiments which we carried out in our lab that we did with both light microscopy as well as a scanning electron microscopy. Now we give some examples of what are all the features you can able to see through the scanning electron microscope that not in light microscope, and also whether it requires scanning electron microscope or the light microscope, that is stereo microscope images is enough. So that is what we are going to discuss now with this. So one is like when you look at here, you can see the bone loss of mice due to periodontal disease as well as diabetic, which require, you can able to see the detailed bone loss. So it doesn't require that scanning electron microscope to visualize this. So the stereo microscope itself, which shows that this area of the bone loss, alveolar bone loss, is clearly we can see. So here also you can see, some of the biofilms forms here, and but we could not able to get a detailed biofilm structure. So if we really wanted to study the detailed structure of the biofilm of any bacteria like a Streptococcus mutans and Candida albicans, that is the core species, we did ex vivo studies. We could not able to see the biofilms, but you can see the, these images very poorly understood. So that means we need a more detailed structure. So the left side, when you look at these structures, is light microscopy. That's a stereo microscopy, which you can able to see the caries lesions in this, tooth. So if we really wanted to see inside how this caries teeth is, things, when you look at here, you can detail the structure of the enamel dentin areas that you can able to see in here in the scanning electron microscope. So this is another scanning electron microscope. You can able to see the crack the teeth that you cannot see in the light microscopy, but you can able to see in the scanning electron microscope. Again, another high magnification of scanning electron micrograph of the smaller one, you can see the structures. So this is Aggregatibacter actinomycetum comitans bacteria which cause localized aggressive periodontitis. which has two different phenotypes. One is smooth colonies, the other one is rough colonies. So the smooth colony is a laboratory variant, which has, it's lacking the forms the biofilm. It's a planktonic cells and free floating when compared to the rough strain that is always isolated from the patients. So when you look at this, the light microscopy, which shows a typical star-shaped structure of AA that lacking in the laboratory variant. When you go for scanning electron microscope, you can see the two different structure of the biofilm of this colony. So this gives the scanning electron microscope have more detail about the overall the structure of this biofilm architecture. So next, what we did is we grow the AA on the surface of the cover glass and took the scanning electron microscope. That is the low power of scanning electron microscope. You can look at this, the mat-like structure of this biofilm forms with attached to this cover glass. It's a low magnification, and when you look at this as a higher magnification, each biofilm is how it looks, a typical architecture of AA. So here also another difference, the biofilm of AA as well as the PG. PG is Porphyromonas gingivalis, that is cause periodontal disease, and you can see the biofilm of different AA and PG. You can typical the differences. So we did with the study on control of the PG, how we can control the PG. So we did an experiment in our laboratory that diallyl sulfide, a compound which is isolated from garlic. So we wanted to study about how important the compound diallyl sulfide, whether that kills the Porphyromonas gingivalis. So when you look at it here, that is control group has low magnification of the Porphyromonas gingivalis. It forms biofilm on the surface of cover glass. When you process the samples for scanning electron microscope, and when you increase the magnification, you can see a typical rod-shaped the bacteria of typical architecture of the PG. When we treated this with the diallyl sulfide for 2 hours, it completely removed the biofilm on the surface, and even the leftover is also shrink and remove the biofilm. Only you can able to see the polysaccharides that is attached on the surface. The higher magnification is also clearly evidence that the difference between the B and D, it's significantly reduced the biofilm, and also most of the surface is free from the biofilm when compared to the control group. That suggests that the scanning electron microscope help us to understand further how this improved the evidence that shows that PG is dramatically removed the biofilm. Similarly, like we did with the Streptococcus mutans and the Candida albicans, dual species, how this forms biofilm on the denture, that also which shows you can able to see a dense biofilm, both PBS treated, and also when you increase the magnification, you can able to see both Candida albicans, yeast cells, as well as the hyphae, and also a lot of Streptococcus mutants. And also when you look at this in the higher magnification, So we can see on the hyphae, the Streptococcus mutans cells is attached, the colony is attached to this hyphae. So hypothetically, we believe that the hyphae is developed only when you immunocompromised. So though it is a commensal pathogen, the Candida albicans. We found in the normal flora, but in yeast form. So once immunocompromised the patient, then it will develop a hyphae, that is a virulent factor that secretes a toxin. So the hyphae is penetrating to the tissues and the cells, and then release the toxin and then degrade that, disintegrate, host DNA and other materials. So if the Streptococcus mutans is also attached onto the biofilm, that also get into the tissues and cause more destruction compared to the single colonies. So this kind of evidence suggests that the scanning electron microscope gives you more detailed structure when compared to the light microscopy. Here also, when you treated with the lactoferrin, you can able to see the difference. It's completely removed the biofilm, and you can see it's a less number of bacteria as well as the fungus. So the brackets, we can see the brackets, we wanted to study about how the brackets are. And, when you have this, we wanted to see how the bacteria forms biofilm on the, brackets. So we studied with the in vitro study on, we incubate these, brackets with the aggregated actinomycetum comitans, and then you can see how the bacteria forms biofilm on the surface of these brackets. So you can see so that also suggests, the detailed structure of the brackets as well as how the bacteria forms biofilm on these surfaces. So another important features that you wanted to compare the light microscopic structure as well as the scanning electron microscope, how this. When you look at this, the light microscopy, when you grow the Candida albicans, it forms a biofilm as well as a hyphae, things. It's a control group, and you can see a low magnification of scanning electron microscope, and you can see it's a, high power, high magnification. Similarly, this scanning light microscopy, when you treated with the human lactoferrin, see significantly reduced the hyphae when compared to control group, and also reduce the biofilm. And you can see here also, you cannot, most of them are yeast form when compared to the control group. That also suggests that the scanning electron microscope gives us a detailed structure and understanding further with the difference between control as well as the human lactoferrin treated cells. So this is another, denture related studies on, denture. So here you can see control groups, and the bottom one is lactoferrin treated. So this is a Streptococcus mutans, and Candida albicans, forms biofilm on the surface of the denture, and this is a dual species. When you look at this, it significantly reduced the, bacterial biofilm and also the fungal biofilm, and you can see we reduced the thickness of these, biofilm when compared to the, this. So that also gives you a, detailed structure of different surface that shows that, the lactoferrin effectively prevent the biofilm. So this is a Streptococcus mutans biofilm on denture with the high magnification. So it's a PBS treated, and whereas you can human lactoferrin treated cells shows the difference between these. Both are took photography with the same magnification. You can see here how it is, and you can see from the two different treatment groups that also you will see the difference of biofilm. Also, the Candida albicans biofilm, which also shows that you can see a typical biofilm of the Candida albicans using the scanning electron microscope. So similarly, we detailed the structure of the co-species biofilm on the denture. You can see a low magnification and a higher magnification. You can detail. So this is a Streptococcus mutans forms biofilm on the surface of denture. And when you treated with the human lactoferrin, the Streptococcus mutans significantly reduced the biofilm on the surface. This is control Candida albicans, and this is Candida albicans. When you treated with the human lactoferrin, it's completely removed the biofilm. And here the Streptococcus mutans and the Candida albicans dual species, and you can see the structure, how the difference is. So this is a typical scanning electron microscope. So here again I detailed this, the higher magnification of the co-species on the denture, and you can see the difference between these two surfaces with the lower magnification. So this is another surface we wanted to compare with the plastic and denture. Now this is on enamel. When you see the co-species forms biofilm on the enamel more when compared to when you treated with the human lactoferrin, that also suggests that this is very interesting and found. So the effect of human lactoferrin on co-species of Streptococcus mutans and Candida albicans biofilm on plastic, denture, and enamel. So here, again, just I give the all three in one, then you can see how the difference is. When you look at this, it's a human lactoferrin treated, groups, and this is PBS treated groups. This is a plastic cover glass on the surface, and this is on the enamel, and this is on the denture surface. So you can see it depends on the surface, the biofilm structure is different. And the treatment group is also, how important on the different surfaces, and the enamel, the Candida albicans and the Streptococcus mutans forms biofilm, and here it's more on the denture and the Candida albicans, and here the plastic, it covers both more, and here more Streptococcus mutans forms more biofilm on the enamel when compared to Candida albicans, whereas in the denture, the Candida albicans forms more biofilm than the Streptococcus mutans. So the surface is very, very important for the bacteria or fungus to form more biofilm on the surface. And also this study, it shows that the differences not only see the biofilm, but also how the treatment group is a response when compared to the control groups. So another elastomere, that is another study which shows that, how important this and so it's 2 weeks after, the elastomere, that is, you can see if, this is like we collect the elastomere from the patients, and then we washed, with the PBS, and then we put them onto the anaerobic chamber. So after 2 weeks, you can see, how severe, the biofilm forms, how dense on the surface of the, elastomere. And this is a control group, so we didn't see any biofilm. So that is also shows that scanning electron microscope gives more things. Again, the same patients we got, the this collected from the patients, and we put them on the media, see how this, microorganism forms biofilm on this surface of the brackets. So this is another interesting, group, we studied, we collaborated with the allergy specialists. They wanted to study about a different pollen. So you can look at this different structure of the pollen. That is another study. And this is another study we wanted to study about, lactoferrin treatment on the heart of mice, how, this will help to remove the AA biofilm on the surface of this, the mice heart. So this is a stent commonly be used for angiogram or angioplasty to remove the block. They use the stent. So one cardiology resident wanted to study about this, want to see how these different companies, stent looks, how after they inserted this in the dog. So we wanted to study how in a different pressure, how these brackets tolerate, when compared to each other. So that is this study. Also, the drug delivery nanoparticles is another important collaborator want to study how the structure of the nanoparticle looks like. Is it good or any damage or anything? So this is a typical nanoparticle. You can able to see lower magnification and higher magnification of these studies. So these are all, important to, the scanning electron microscope. So here we not only study biological samples, but also like drug nanoparticles, stents, and then, other things. So as I mentioned during the process, this is a metal, silver, but my question to you, whether this require fixation or not. As I mentioned earlier, that if there is no bacteria you are looking for, then no need for fixation. But if you are looking for the biofilm, then you need to fix the samples with osmium tetroxide or glutaraldehyde, dehydrate these, and then drying, critical point drying, and then coat the samples and took the pictures. This is a typical example. But for this, you no need for process your sample. Just directly you coat the samples and observe. Whereas for this kind of study also, it require fixation, dehydration, drying, and mounting on the stub, and coating samples observation. So these all, important aspects of, things. So now you understand that how important the scanning electron microscope and what are all the informations you are going to get that does not get it from the light microscope. So how efficient this and how you can improve the quality of your samples. Also, you can get the details of different materials, not only just biological samples, but you can get anything you want. So with this, we will complete the scanning electron microscope. So far we completed the light microscopy, scanning electron microscopy, fluorescence microscopy, laser capture microdissection, and all these process. So as always, important to note that the basic principles of each microscopy and how does important and what kind of data you are getting, what kind of structure you are getting from each one, and the process, whether the bright field microscopy and fluorescence microscopy, the process whether same or not, at what point you are changing the process, and also not only you observe the structure of the samples, but also you can able to isolate the cells. Okay, once you isolate the cells, what are you going to do with this? So these all the informations we discussed about this. So any students, if you are interested to come in person, let me know. I will show you the microscopes. So interested, and let me know so that you can, I will show you all these instrument and how it does work and everything. Okay. So usually I do in-person hybrid this, and so always welcome. And let me know your things. I will ready to help you to show all the works. Okay. So if everything is okay, if you need any questions, anything, feel free to reach out to me. Do you have any questions right now?